पवन। हेलो। हेलो पवन।
क्या?
टॉपिक बायो में क्या हो रहा है, कुछ पता है सर क्या करवा रहे हैं?
बायोटेक्नोलॉजी करा रहे हैं अभी।
ओके, थैंक यू। सर कब तक आएंगे? आ ही नहीं रहे हैं 6:30 से।
पता नहीं।
ओके, थैंक यू।
Good evening.
Good evening, sir.
Good evening, sir.
Yeah.
गुड इवनिंग सर।
Yeah, very good evening.
कैसे हैं सर आप?
मैं ठीक हूँ, आप कैसे हो?
मैं भी ठीक हूँ। सर मैम की तबीयत कैसी है?
या अभी ठीक हूँ। अभी
ओके
अच्छा।
सर।
Yeah, see here. So in yesterday's class, we have discussed this genetic engineering, then the techniques, okay, how to maintain this chemical engineering process, microbial contamination free. Then we need to produce large quantities of the desired product, and here the product may be antibiotic or vaccine or enzymes, etc. Okay. And here, from the beginning, we are discussing that a desired DNA can be cut out, from the human genome. Then we can combine with this plasmid like this in the presence of ligase. Then here, so few points are here. See. First, recombinant DNA construction. What about the work of this, Boyer and also Stanley Cohen? See, construction of artificial recombinant DNA molecule. How this, first thing came into existence? You see here, emergence of first recombinant DNA construction. Then we discussed that this Stanley Cohen and also Herbert Boyer in the year 1972. What they did, they isolated antibiotic resistance gene. From whom? By cutting out a piece of DNA from the plasmid. And here, they isolated this particular antibiotic resistance gene. This antibiotic resistance gene is present in the plasmid. This is the plasmid. So somewhere, this antibiotic resistance gene is present. Suppose this is the antibiotic resistance gene. And where is the location of this plasmid? This plasmid is seen in the case of bacteria like this. Suppose if antibiotic resistance gene is present in this plasmid in this bacteria, so what will happen to this bacteria? Even if you give this antibiotic, and this bacteria will not die, because this plasmid is having a gene which is not yielding to this antibiotic. That is what is antibiotic resistance gene. Sometimes what will happen? In the TB, in the diseases we will discuss. In the TB, if you use the antibiotics as per your wish, then antibiotic will not work, second time, third time, maybe 20th time. Because meanwhile, the organism will develop resistance. How it
Now,
will
what is
develop?
TB?
Yeah, in TB that is going on. In TB, tuberculosis, okay.
Okay.
in TB, tuberculosis, antibiotic resistance is a common factor. That is the issue. The individual, even though he take antibiotic, the individual will suffer from the Mycobacterium tuberculosis. Mycobacterium was the bacterium. And this Mycobacterium will not die with this antibiotic. Why? What happened? Because this Mycobacterium developed resistance to the antibiotic. Why? Because there may be a gene. So here what this Stanley Cohen and Herbert Boyer did in the year 1972, there is a gene which is called as antibiotic resistance gene that is present in the plasmid, that was cut down. That was separated. Now this gene, which is nothing but antibiotic resistance gene, that is separated from this plasmid. Okay. This gene confers antibiotic resistance. Okay. So these people, they isolated this antibiotic resistance gene from the plasmid of one bacteria. The cut piece of the DNA then linked with the plasmid DNA. Now this is cut down and take another plasmid from another bacteria. Okay. Now this is the antibiotic resistance gene. Now this gene is linked with this plasmid. That is what they did. This antibiotic resistance gene is here now. Okay. These plasmid DNA act as vectors. That is what we discussed. Now this is acting as vector. Okay. Why? This vector is transmitting this gene, which is not present in the plasmid previously. This gene is present in some other plasmid that was cut down, then ligated here. Now this is called as vector. Okay. Now, what happened? This plasmid can be used as a vector and it can enter into any other bacteria. This is what is called as host bacteria. We are discussing the same from the beginning. Okay. Why this is called as vector? Because like the mosquito, Anopheles mosquito will act as vector for this malarial parasite. That is why in that sense it was named as vector. Then you see this ligase is important in ligating these molecules. And here this ligase linked the antibiotic resistance gene with the plasmid vector, and this became possible with the ligase. This makes a new combination. What is that new combination? This recombinant DNA produced because of this new combination, because this antibiotic resistance gene from one plasmid and this is from another plasmid, recombinant DNA is produced. Now see. See here. Okay. Now you say the names of the people, names of the bacteria here. See. When this DNA is transferred into this Escherichia coli bacterium, and which is closely related to this Salmonella. Okay, what is this Salmonella? From this particular Salmonella only, this antibiotic resistance gene was isolated. Now this newly formed recombinant DNA was added into this Escherichia coli bacteria, E. coli bacteria, and it could replicate because the same type of plasmid can be introduced, and multiple copies were made in this. So the ability to multiply copies of antibiotic resistance gene in E. coli was called cloning of antibiotic resistance gene in E. coli. Now what happened in this? What happened in this? See, one bacteria, this is the bacteria, And here, this is the plasmid which is there in bacteria. This bacteria is what is called as Salmonella. You know that typhoid is because of this Salmonella only. Salmonella typhi and Salmonella paratyphi are responsible for this typhoid. And from this, so there is one gene called antibiotic resistance gene, and this was isolated by using the restriction enzymes. Now, who is this useless fellow? Huh? Who is doing this?
Sir, someone has joined with my name and do this. Pavan. I don't know, sir, who is this.
Pavan? See, I can
Sir,
take out.
sir, now he is left the meeting.
Who is this Pavan?
Sir, someone has joined with my name.
Acha?
Yes, sir, and do that. I don't know, sir, who is this. Sir, he changed his name again and again.
Okay. We'll see that. But don't send, from tomorrow onwards, we will send only to the mails. We will not keep this in the group.
Okay, sir.
Okay. See here, this Salmonella, from the Salmonella, this antibiotic resistance gene can be taken out. Then this will be added to one more plasmid. And this resistance gene was added. And it will kept in another bacteria. That bacteria is what is called as E. coli. Now, what E. coli will do? E. coli will have this antibiotic resistance hitherto, which is not having this. That means the character which is present in the Salmonella is now seen in E. coli because of the presence of this antibiotic resistance gene. This is what they did. Stanley Cohen and Herbert Boyer. Is it clear?
Yes, sir.
Yeah. Now,
Yes, sir.
yeah, in the genetically modifying organism, three basic steps are there. What are those three basic steps? Identification of the DNA with the desirable genes, then introduction of identified DNA into the host, and maintenance of this introduced DNA in the host and transfer of the DNA into the progeny of this bacteria. And if this bacteria multiply, what will happen? Automatically this gene will be transmitted to the progeny. Progeny is nothing but offsprings. So you need to identify the desirable gene. This is what is a desirable gene. You need to identify. And you have to introduce into this, vector. Okay. And into the host. Host is nothing but another bacteria. E. coli is now host. These are the basic steps. What are the two important bacteria you have to remember in relation to this, Cohen and Herbert Boyer? Salmonella, then E. coli. Is it clear?
Yes,
What
sir.
is bacteria?
Yes, sir.
Typhoid is caused by Salmonella typhi and paratyphi. And tuberculosis is caused by
Mycobacterium.
Mycobacterium tuberculosis. Mycobacterium tuberculosis. In yesterday's class, we have discussed that we will isolate the DNA material by giving different enzymes. We need to digest the proteins, we need to digest the lipids, we need to digest the carbohydrates, we need to digest the RNA. And here from the bacterial cells, we need to take out this DNA by lysozyme. Then plant cells can be digested with cellulase, and fungus can be digested with chitinase. Okay. See here, after this, we discuss this agarose gel electrophoresis. Agarose gel electrophoresis, because this is the negatively charged one, it will go to the anode. And the same process can be repeated for vector DNA even. And here this restriction enzyme will digest and it will produce a number of fragments like this. And these fragments, because of the negative charge, they will move to the positive charge. Okay. And how they will move, we need to see. Okay. And who is this Shilpa?
Sir, now he is just changing his name Pavan to Shilpa.
He joined with the name of Priyanka, as there are two Priyanka in class. No, sir, Shilpa.
Sir, Shilpa left the meeting now.
Sir, you can log the meeting after we joined it. So no one can go outside of the class or can in the class.
Yes, sir.
As if you send on mails, the person can also join it by mail. Sir, Mohammed Sakir is doing this. His name is shown on screen now.
Who is this person?
No, sir, मैंने नहीं किया.
Your name shown on the screen.
मैंने कुछ नहीं किया.
What nonsense they are doing? Who is these two Priyankas?
Yes, sir.
One Priyanka left now. Ray, my dear, you will not be eligible to write any examination in the country. Don't play with us, useless fellow. Who is this Priyanka? Who is this person? Where is Priyanka?
She has left.
Ray, what is going on? These people are disturbing. Okay. So in yesterday's class, we have discussed the different, things, how to produce this polymerases, chain reaction, then how they will produce the desirable DNA in multiple copies, etc., etc. All these things. So this we have not discussed. Once the recombinant DNA is prepared, there are several methods to introduce into the host cells. So once the host is ready, then it will produce the desired gene product. How to produce the gene product? That is the challenge. How to produce this gene product? Gene product, we have given some gene, and that gene is now in the host, and that host has to produce this product. So for the host, this gene is nothing but a foreign gene. This foreign gene gets expressed under appropriate conditions. That means you need to provide this appropriate condition. What are those appropriate conditions and where these appropriate conditions can be put in? Okay. So here, the culture can be given to this host. The culture is nothing but for the survival of this host bacteria. Cultures are essential. That is the nutrition for this host. So this host has to produce this desired protein by taking this nutrition. That is why culture must be produced. So this medium, okay, and this medium must be drained out from one side, and well, fresh medium should be added from the other side. Suppose this is the reactor which can, and here the medium can be given like this, and the medium will goes like this. And meanwhile, this is the bacteria host, and that will be utilized this medium. And that is how this bioreactor will be. See this. This is the reactor. See, this is the reactor. This is simple stirred tank bioreactor. And in this tank bioreactor, okay. And here is, because the diagrams are given, maybe given, and hence you should know what are the different parts and other things. For pH control, this is pH control. This is pH control. To maintain this pH control, because appropriate conditions are essential and they can be provided in this bioreactor, which is stirred. Stirred means it can stir, it can mix all the contents. So here, the steam can be provided here for the sterilization purpose. We discussed that in the chemical engineering process, the ambient environment must be created. This is no bacteria, other bacteria must be present in this other than the host. And hence, this sterilization is essential. For that particular purpose, the steam can be provided. Okay. Then later on, the foam breaker, foam will come, and that will be, the foam will be broken by these, things which are fan-like things which are present here. And the culture broth can be kept in this. This yellow color is nothing but the culture broth. Culture broth. Then this flat bladed impeller, and this will mix throughout. And the sterile air can be provided. And this is the motor. And this is the bioreactor. So the culture broth is very, very important. This culture broth is nothing but this is the culture medium. And this bacteria will consume this and will multiply. And will multiply, the number of progeny will be seen. And the progeny will also have the desirable DNA. And the progeny will produce this desired gene product. And that can be collected. How to collect? See, this is one more sterile, bioreactor. Okay, this is stirred bioreactor, tank bioreactor, big tank. Then gas entrainment, gas will be present here. The surface area will be increased. Okay, and here this will spray. The sparged. In this, the sterile air bubbles are sparged. Sparged means sprayed. Okay, the surface area can be increased like this. And here, the oxygen, the bubbles dramatically increase the oxygen transfer area. So this is the arrangement which is present in this, bubbled, sparged bioreactor. In this bioreactor, you see, in this bioreactor, it provides, what it provides? See, optimal conditions for achieving the desired product. Optimum growth conditions. That means temperature, pH, substrate. Substrate means the culture. And based on the substrate, the bacteria will. Then salts, then vitamins, then oxygen. All these things will be provided to this bioreactor. Because large scale production is essential. So to produce large scale productions, then bioreactors are helpful. And here, you see this. A stirred tank bioreactor is usually, with this cylindrical or with a curved base. For what purpose it is cylindrical? For the mixing purpose. And here, stirrer, what the stirrer will do, it will mix properly. And so that the oxygen will be available throughout the bioreactor. That is the purpose of the stirring. And here the air can be bubbled throughout the reactor. That is the purpose of the stirred tank bioreactor. Okay. And here, after this, the downstreaming process, we discussed this downstreaming process. What about this downstreaming process? After production, what you should do? There are a number of series of processes after biosynthetic stage. Biosynthetic stage. Synthesis is over. In the bacteria, the concerned insulin may be produced. So you need to separate, then you need to purify, then you need to add preservatives, then quality control testing for each product. Okay, then this is what is the downstreaming process, so that the quality can be maintained. And here this will vary, the control system will vary, the quality control system will vary from one product to the other product. So what are the different steps which are there in downstream processing? You need to separate the insulin from this bacteria. That is what is separation. Then after separation, then you need to add the purifiers. Then you need to add these preservatives for the purification purpose. Then quality control testing. This is what you should do in the downstream processing. Is it clear? This is
Yes,
how
sir, clear.
Yes, sir.
the gene product will be produced. Okay. Tomorrow you see this, mail. Who is this Palash? We'll see this tomorrow. Right? So what is the next chapter you want after this? We will go for application. And after applications, what chapter you want? To please tell me.
Sir, human health and disease.
Sir, human
Human
health
health and
disease.
disease.
Health and disease. Okay, fine.
Yes, sir.
Okay. We'll go for human health and disease after these applications, right?
Okay, sir.
Okay, sir.
So tomorrow we will see this, right?
Sir, if we have any doubt in this chapter, so we can ask.
Yeah, you can ask.
Okay, sir.
You can ask. No issue. Who is this Muhammad Sakir? Yes, Sakir.
सर
Huh?
सर आपने मेरे को मीटिंग से रिमूव कर दिया था, मैंने तो कुछ करा नहीं था, किसी और ने मेरे नाम से ज्वाइन करा था, प्रियंका के नाम से।
What, Priyanka?
सर किसी और ने ज्वाइन करा था मेरे नाम से।
I
Sir,
don't know.
she's saying some other person joined with her name.
Yeah. I understand.
Sir,
Yeah.
Rakshi is saying that please don't send the links on mail because he has given her mother mail, so her mother phone is different and her phone is different. So please send the link on group only.
Group, it is not possible. Because these disturbances are going on.
But sir, on the mail also it would be happen now. Like because only group students are doing this.
Okay.
Yes, sir. Same thing will be
Sir,
happened in
if you mail,
class also.
the person will automatically get the link as he present in group.
सर you can change the setting of meeting.
How?
Yes, sir, you can log the meeting.
Sir, there is setting only host can share the screen and make changes.
Yes, sir.
Acha, log the meeting.
Yes, sir.
Tomorrow we'll do that. Right. Right.
Thank you, sir. Good evening, sir.
Right. Good night.
Thank you, sir.
Thank
Bye,
you,
sir.
sir.